All right, we're just going to wait another 30 seconds or so as more people come in. All right, let's go ahead and get started. Hello, everyone. Thank you for joining us today for the 5th webinar in our EPIC webinar series on the use of NAMs and risk assessment. This series is being co-organized by the Environmental Protection Agency, PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. I'm Rachel Bruner, a biologist with the US EPA, and I'll be co-moderating today's webinar along with several others, including Naladri Baumick, a toxicologist in the Human Health Assessment Branch of the California DPR, Amy Clippinger, president of the PETA Science Consortium International, and Christy Sullivan, the vice president of education and outreach at IIVS. Before I introduce our speakers, note that the slides and a recording from today's webinar will be posted online at this link shortly after the webinar. All future EPIC webinar materials will also be available at this link as the series continues. We have 2 speakers today who will be presenting on the CATMoS model for acute oral toxicity and evaluation of its potential for use in a regulatory context for pesticide, hazard, and risk assessment. First, we'll hear from Kamel Mansouri from the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM, which is located within the U.S. National Institute of Environmental Health Sciences. And second, from Michael Lowit from the U.S. Environmental Protection Agency, Office of Pesticide Programs, the Environmental Fate and Effects Division. We will also do a Q&A session at the end of the second presentation. The webinar is scheduled for 1.5 hours, and we may wrap up earlier, sort of depending on how long the questions go. Everyone is muted. You can type any questions in the question module within your Zoom toolbar at the bottom of your screen. And go ahead and please indicate if your question is directed to a specific speaker. We just ask that you don't add questions to the chat, just to the Q&A module. We've also enabled closed captions, and you can turn this feature on or off according to your preference using the toolbar at the bottom of your screen. With that, again, thank you to everyone for joining today and engaging in this topic. And thank you to our 2 speakers, Dr. Kamel Mansouri and Dr. Michael Lowit, for preparing for and speaking for us today. Thank you to our co-organizers, PETA and IIVS. They do so much of the behind the scenes work to keep these EPIC webinars up and running. It is much appreciated. I'm hearing a little bit of background noise. Thank you. And I really look forward to seeing how CATMoS evolves as a tool to aid in risk assessment of acute toxicity in the future. Now our colleague from the California DPR, Naladri Baumick, will share some comments.
Hi, Rachel. Thank you. And can you hear me?
Yes, we can hear you.
Yeah, and there will be some background noise, so I will cut it short. I'm currently in India, and my home is right behind the street. So Please bear with me. Anyway, hello and greetings to everyone. And on behalf of everyone at California Department of Pesticide Regulation, I cordially welcome you all to one more edition of the EPIC webinar on NAMs. We, particularly at CDPR, are excited to be part of this webinar series, along with our partners US EPA, IIVS, and PETA. Personally, I'm extremely privileged to be part of this panelist team, which includes members who are continuously making NAMs meaningful and effective. To our speakers today, I express my sincere gratitude to you and extend our warm welcome. Like everyone else here, I'm also eagerly waiting to listen to presentations on CATMoS model for assessing oral toxicity, especially important because of its potential use in regulatory hazards and risk assessment. With those words, I welcome you again and return it back to Rachel.
Thank you, Niladri. So now, before we introduce our first speaker, we're going to try something new. Throughout the program, we're going to ask you, our audience, for input a couple of times to try to understand the expertise in the room and your views. So we have a few polls. We've got our first 3 poll questions here. Poll question number 1, on a scale of 1 through 5, how much experience do you have with QSAR modeling in general? 0 is none at all, to 5 is a lot of experience. Poll question number 2, on a scale of 1 to 5, how much experience do you have with the CATMoS model specifically? And question number 3, if you have used CATMoS, was it for exploration or just sort of learning about the tool? Was it for research or internal decision making or for regulatory submissions? I'm going to give you some time to answer these questions. All right. While you continue to give us your input, if you haven't done so already, I'm going to go ahead and introduce our first speaker for today, Dr. Kamel Mansouri. Dr. Kamel Mansouri earned his PhD in computational chemistry from the University of Milan in Italy as a Marie Curie Fellow. Since 2020, he has been spearheading computational chemistry endeavors at NICEDM within the U.S. National Institute of Environmental Health Sciences, or NIEHS. His focus includes QSAR modeling, cheminformatics, and computational toxicology, with a reputation for fostering international collaborations and leading scientific consortia. In 2017, he was honored with the Lush Prize for pioneering in silico alternatives to animal testing for screening endocrine disruptors. All right. Let's take a look at the poll results. Interesting. Thank you for your input so far. Let's get started with the presentations. Kamel, I see your presentation, so please go ahead.
Thanks a lot, Rachel, for the introduction, and thank you to the organizers for the invite to share a little bit about CATMoS. And thanks everyone who joined online. I see almost 300 people online, so that's amazing. Thank you all for being here. And thanks also again to the organizers for putting the poll before starting. That gives us an idea how to orient a little bit the talk and make sure that it's beneficial to you. So with most people, there's higher number of people who have no experience with QSARs and with CATMoS in general. I will not be very technical, so I'll go quickly through the technical parts of the slides, and then I'll try to give a short demo about the tool, just in general. And then at the end, I'll be happy to answer any questions, either about the tool itself or about QSARs at the end of it. So CATMoS is a QSAR model that predicts chemical properties, just like any other QSAR model. It starts with the chemical structure, and it trains a model based on a lot of descriptors derived from the QSAR, from the chemical structures, and tries to relate them to the activity being modeled and predicted. So it uses existing data, existing experimental data, to build the model and apply it to new chemicals. And that saves a lot of time, costs, and of course, the animal welfare. So CATMoS was a third of two other collaborations organized by the EPA and IIVM. Specifically for CATMoS, it was the acute toxicity work group within the IIVM. And before that, there was the estrogen receptor activity prediction model, and also COMPARA, which is about the androgen receptor activity. Those two are within the EDSP program, the endocrine disruptor screening program at the EPA, and CATMoS for acute oral toxicity. And coming next, we're currently working on acute inhalation toxicity. So I'll keep you posted on that. So this international collaboration, like I said, it was the third collaboration after the successful first two. And it involved over 100 scientists from around the world. And we had a publication about CATMoS that I invite you all to take a look at. You will learn quite a bit about the model itself and how it was built. So today we won't be focusing on that. And this is the list of all co-authors, as you can see. And these are the countries that participated, where the groups are from. So CATMoS was built specifically to address certain needs. And prior to that, we tried to make sure that we are aware and also address those specific needs of the regulatory agencies. So we worked closely together with them from the beginning. And that's how we were able to build CATMoS, this suite of models. And the reason why we call it a suite of model, not a single model, it's because it's actually 5 different models bundled together in this suite called CATMoS. So CATMoS actually predicts the point estimates, LD50 values, but also other endpoints like highly toxic versus toxic, or toxic versus non-toxic, as well as multi-categorical endpoints like the EPA categories, 4 categories, and the GHS 5 categories. The data set that was used for the model was about 12,000 chemical structures. And it started from a lot more than that, and there was a lot of curation involved. A big team of curators worked on preparing this data set. So kudos to everyone, and thanks to everyone who participated in that, who prepared the data sets for us. And then after that, we split that initial database into the 5 endpoints. That's why you see different numbers for each one of the endpoints, because it depends on the availability of the data, like whether it's continuous LD50 values or just range that can be used as binary or multi-categorical data for training models. So the models were trained separately. And that's what we gave all the participants, those 5 data sets, separate data sets. Obviously, we split them into a training set that took 75% of the data and an evaluation set that took 25% of the data that was kept hidden from the modeling groups to allow for evaluation later on and making sure that all models are functioning correctly. The splitting of the data obviously took into account the distribution of the data sets and the quality of the data points that were included in each, the training and the evaluation set. The evaluation set was included into another bigger list that we call the prediction set that we gave all the modeling groups to make predictions after they build their models. So it was about 50,000 chemical structures that included the evaluation set. And the reason we did this is first to have the separate model predictions on lists of interest to the regulatory agencies that we work with, but also to include the evaluation set within those 50,000 so that we can evaluate the model. But obviously that was not to compare the models, which is better or to rank them in any way. It's more to build the best consensus model so that we get the best consensus predictions at the end. And as you can see here, you have the 5 different endpoints, and we receive different models for each one of the endpoints, depending on the interest of the modeling groups and their experience with the way they apply their approaches. And in general, all the models were very highly concordant, meaning they agree on which chemicals are toxic and which chemicals are non-toxic for the most part. The other important thing is that the models were covering most of the prediction set, the 50,000 prediction set chemical structures. And that means that those structures were within the applicability domain of the models for most of them. So as you can see on the left-hand figure, most chemicals had coverage of between, let's say, 18 and 25 models, but a minimum of 10 models. So each chemical was at least in the applicability domain of 10 different models. And that gives us this is very important, especially for consensus models, so that it gives us the confidence that we need to combine those models into a consensus, statistically speaking. After that, as I mentioned, all the different participants provided separate models for each one of those endpoints, right? So we need to combine them into a consensus that is consistent. So first, we combined each endpoint separately, the models from each one of the 5 endpoints into a consensus model per endpoint. So that, like, for example, the LD50 25 models, we ended up with one single LD50 model. So that's the first step. After the first step, we wanted to make sure that our CATMoS, when it predicts an LD50 of certain value, it falls within the right EPA and GHS categories, for example. So that's where we developed the second step, which is the weight of evidence approach that took the majority role. And we did a little bit of adjustments, obviously, because those models being developed separately for each endpoint, sometimes they differ a little bit. So we made adjustments so that the final output, the user, whether they look at an LD50 value or an EPA or GHS category, they have the same prediction, and it's all consistent. After that, we evaluated the consensus model statistics, and the important take-home message is that the predictions were pretty accurate in comparison to the repeatability of the in vivo data. So as you all know, there is some variability within the in vivo data that at least when you repeat a chemical, like an experimental assay for one of the chemicals, you might get a little bit of different values for LD50, for example. We tried to be within that range. And we applied that range of variability to the consensus predictions at the end. So, and I'll show you later what I mean by that. After that type of validation, we also worked with other agencies to further validate and learn a little bit more about how the model behaves for specific data sets and specific groups of chemicals of interest to each one of the collaborators. And later on, Michael will talk to you a little bit more in details about one of these efforts that is led by Patricia Bishop, and this is the publication that just came out this year that I invite you all also to take a look at, and Michael will explain it a little bit more in detail. So a model is just a model on paper that does nothing, in my opinion, unless it is implemented and provided to the users as an application so that they can apply it on new chemicals and get all the information that they need on their chemicals that they're getting predictions from. So all those models that I mentioned previously are included in this tool called Opera that is free and open source. And it is built, like all the QSAR models included in Opera are built on the 5 OECD principles for QSAR validation, so that all the predictions are adequate for to help with the regulatory processes. So Opera pipeline takes very thorough steps to curate the data sets that we use. All the code that is included in Opera is open source, and all the code is provided on the GitHub repository of Opera within the official NIHS GitHub repository. The algorithms are also unambiguous and transparent, and the tool also provides the applicability domain and the limitations of the different models, as well as the accuracy estimates for each one of the predictions. And also, you can also access the predictions for certain models within the Nicetum ICE web tool, and some of them also within the EPA's ComTox dashboard. So this is how Opera looks. So it's provided in 2 main different flavors. One as a command line for programmers so that they can embed it within other tools or query it in the background without having to open the interface. And most people use the user-friendly graphical interface that's much easier to interact with. And that's what we're going to focus on today. So Opera provide not only the model CATMoS, like I mentioned, but also the other 2 collaborative models, the SIRAP and Compara, as well as other physical properties, environmental fate, and ADME properties. And I'll talk a little bit more about the input options that can be chemical structures in different formats, or even structure IDs to facilitate the use of the tool itself. The tool is available for Windows and Linux, and we're working on a version for Mac OS. And there are certain libraries in other languages to embed the tool within other tools as well. One thing to note is that the models are being always updated. So there's a different versioning for certain models in comparison to the tool itself. Sometimes we update the interface and add new features without touching the models. And sometimes we provide that update with updating certain models as well. But we focus on the certain lists of or classes of chemicals of interest so that our models are always accurate. Now, Opera is also available on the ECD or the OECD QSAR toolbox, so that with the right plugin, you can get the predictions within the tool itself and compare it to other models as well. So Opera currently was downloaded over 8,000 times from its GitHub repository. And I'll show you a little bit on how to get it running and all that. So you go to the GitHub repository for Opera, and you download the specific file that you need for your OS version and all that. So just before moving to that part of the demo, I wanted to show that how to access Opera predictions from ICE. You can access the different models and download the predictions for your chemicals using your identifiers without having to run the tool, for example. So getting to the demo, there was a demo that was recorded a couple years ago. It's available on YouTube. I don't know if this one will be available on YouTube as well, but you can check that demo as well on YouTube. And we're gonna try to do more or less the same thing here today in a live way. So let's see. Okay, let's go here. So this is the GitHub repository, the official GitHub repository of Opera. Here you have the code, some information, the install guide, some other information. And these are the references of the different models that were published over the years, and we're working on a new one. And this is the list of all the models that are included in Opera. But for you as a user, you don't need any of that. You just go directly to the release here. And this is where you see the latest release that you will need to look at. So there's a little bit of intro about whenever there's an update or certain concerns being addressed. So you skip that part. And then this is one of the most important parts here. And this is where it tells you which file to download based on your operating system that you have on your computer. So whether you want the graphical interface or the command line or the parallel version, whether you want the Linux or Windows version, and based on this intro here, you decide what to download. And this is the list of updates, just a little bit of history. And then this part is also important. It talks about the tool requirements and what you need to have installed on your computer before being able to run Opera. So obviously you need Java. You most probably already have it on your computer if you're running other Java applications. Otherwise, you will need to install it. And then there are certain limitations that applies to most QSAR models. Specifically here, we only predict organic chemicals, meaning mixtures and inorganics and metalloorganics. They don't work here. And there is a maximum molecular weight, like proteins and peptides and nanomaterials. They're not going to be you're not going to be able to predict them. And this is the list here of the files that I mentioned earlier. For example, these are the different libraries, and this is the UI, the user interface for Linux, and this one is for Windows. So it says targz, that's for Linux, and zip, that's for Windows. And this is most of the time, this is what you're gonna download, right? So when you download that, this is what you're gonna get after unzipping the file. You're gonna get the installer and a little bit of information with that. So this is what you run to install the tool on your computer. This is the list of the models and their versions, and you can see also units, and some of them have QMRF reports that give you all the details of the model itself and what's related to the model. I'll invite you to take a look at the install guide because it tells a lot of information about the tool. So this is what I just explained. You go to releases, you install the file, you run the installer, and then it guides you through the installation. It's very simple and quick. You just click next, and you put where you want to install it, click next, and then you have the tool already installed. After that, you can put a shortcut on your desktop, and whether you downloaded the command line or the graphical interface, it's going to be the same. So you'll have the command line and the graphical interface, and that's how they look. Now to the files themselves. So as I mentioned earlier, you have different options for input. So either a SMILES file, and I think this will be most of the time, this is what people use, SMILES structures. But there are other options as well, like SDF and MOL files. And then we added another option, which is a text file containing identifiers, so that if you don't have the chemical structures, you don't need to look for them if you're not sure. You just put the identifiers in a text file, .txt. Remember, the SMILES is .smi. That's how you make, and I'll show you a little bit how to make one. But this is also another easy option so that you can get the predictions without having to grab the chemical structures. And for the organizer, please let me know when I reach my time, because this is, the demo is the last thing in the presentation. You input the identifiers that can be KSRN, DSS Tox ID, or INSHI, and you can also mix them together. I'll show you an example of how to do that. And then another important thing is that if you start from structures and not identifiers, it's better to keep the standardizer option on. That will standardize the chemical structures in the same way, in the same type of representation for the tautomerization and all the structural related information to the way the training set was prepared, so that you get the most consistent predictions. And this is a little bit of information about the QSAR-ready standardization workflow that was just published. So these are a little bit of options on how to use the command line for anyone who's interested. There are examples, and you can get help also through the command line for anyone who's interested in the command line. And these are just some examples. I will not go through them. You have that in the PDF file. Okay, so, and then I'll show you also an output. You can get the predictions in different output formats, but we'll focus about what most people use, which is the Excel or the CSV, right? Yeah, so this is Opera. This is the tool itself. So like I said, when you download it, it will come with sample files to run. So the sample files, this is how they look like. This is a smiles file. It's .smi. It's just a regular text file. I'm using Notepad++, but you can also use the regular Windows Notepad. It's gonna do the same thing. So and this is a sample of the identifiers. It's .txt, right? So one thing most people do is let's close this one for now, is that they have a file like this, right? You starting from a CSV or an Excel file, you have your structures, you have your smiles here, and you want to make a prediction. And you want to create your smiles file from scratch because you don't have a smiles file. So it's very easy. I just copied here just a sample out of these. The important is that you copy the smiles, right? So you take that column and you can copy it to a new text file. And then the important thing is that there should be no empty rows in here. So if you run this, it's not gonna work. The smiles is standardized. You cannot have an empty row. So you remove that. You remove this last one, and then you save it as epic.smi, right? And then you save it.
Do you want to say about 4 or 5 more minutes?
Okay. Yeah, so just real quick. So you select that input and you click calculate. So it will first standardize, create the QSAR ready structures, and then it will calculate the molecular descriptors, and then get the predictions out of those structures. And it only takes a few seconds, like 20 seconds. Then you have your predictions in here in the same folder, and these are the predictions. So because we only included smiles in the input file, it just added like some identifiers. It's gonna be the same sorting in the same order of the chemicals. And these are the different 5 endpoints that I mentioned. This is the LD50, and this is the range based on the uncertainty around each one of those predictions. And this is the applicability domain index, and then confidence index. This is the accuracy for each one of those predictions. Some of them are gonna be high, some of them are gonna be a little bit low, and it depends. So one other thing that I think would be important to mention here is that when you have CAS numbers that you want to include in your smiles file, right? So like this is not gonna work. So if the CAS is before the smile structures, that's not the right order. You will have to have the smiles before the CAS number or any other identifier. You can even put molecule 1, molecule 2, 3, 4, you know. So when you take them together like this from your Excel file and you put them in a text file like this, it's gonna paste it in the right format so that you have a tab delimiter between the chemical structure and the CAS or the identifier. So they have to be tab delimited. This is one tab between each one of them, and that's how you run it. That's how you make a smiles file. If you don't have the smiles file, you can also create that using just the identifiers, and you go to your notepad, you save it as epic.txt, and you run that using Opera, epic.txt. It will generate the smiles for you and calculate it. So you see, it already identified, like you have 17 identifiers in your input file, and it's very quick because it doesn't go through the standardization. So it's only 6 seconds for those, and you get it creates the smiles for you. So that's a plus, and it gets the predictions out of that input file. You have also other options here. You can get the experimental values, the nearest neighbors. The experimental values will work only if you have cast numbers or some identifiers in the file, so that it links the experimental values, if available, to those identifiers. How am I doing on time? Is it done?
Maybe about 1 more minute if you need it.
Okay, thank you. So let's see. Yeah, so one more thing that came sometimes is that sometimes if the cast number is wrong, or it doesn't work, or we don't have it in our data set, Opera will tell you that, hey, we don't have that one specifically, and it will tell you why. So let's take this file, for example. It has 52. It originally had 50, but I included these two on purpose. And so it's getting those structures, right? But it will not find, like, it will not predict those two. And it will tell you exactly why. So in this folder, in the same folder, it gives you all the information related to it. So it's ended 16 seconds. Okay, perfect. Let's take a look at the output file. So you see the input was all these identifiers, and you see their mixed cast numbers, DSS tox ID, and it tells you, oh, I found this one by cast. This one is the DSS tox ID. This one is an issue key. So it identifies all those. However, the last two, this one is not found. It did not recognize it. It's not in our internal database. And this one here, it recognized it, but you cannot predict it because it's inorganic. So it will tell you that. It will not get you predictions for those. And it will predict everything else. So I think that's it for this. Yeah, thank you everyone for your attention. And this is our amazing team. This is the latest picture of 2024. These are not the same people. Like, some of them worked on the project, but some of the people who worked in this project, they went to other careers now. So I want to thank everyone also who participated in these projects, all our international collaborators, our EPA, our IIVM collaborators, and that's our mailing list if you want to keep informed about our latest and greatest. Thank you all.
Thank you, Kamel. That was a great presentation. Not only was it like a great intro to CATMoS, it was also like getting into the details of how to actually use it. So I'm sure that's very helpful for a lot of our audience. We are going to hold our questions to the end of the presentation, but please add them as they come up to the Q&A module. And again, note if they're for a specific speaker. Before we get to our next speaker, we have another poll. Thinking about QSAR models in general, have you used them to make predictions for fish, birds, mammals, other aquatic organisms, or for chemical property predictions. And as you answer those, I'll introduce our next speaker, Dr. Michael Lowit. Dr. Lowit is a senior science advisor at the US EPA Office of Pesticide Programs, Environmental Fate and Effects Division, otherwise known as EFED. He's worked in EFED for 15 years, conducting ecological risk assessments in support of regulatory decisions on the registration of conventional pesticides. He's involved in EPA's efforts with new approach methodologies and currently represents the Office of Pesticide Programs on the Ecotoxicity Work Group of the Interagency Coordinating Committee on the Validation of Alternative Methods, also known as ICVAM, and the Validation Management Group for Ecotoxicity Testing of the Organization of Economic Cooperation and Development, or OECD, Test Guidelines Program. All right, thanks again for your input, everyone. This is really, really helpful for our speakers. Michael, I see you're in, I can see your PowerPoint, but it's not in presentation mode.
Okay, I tried. Y'all are gonna have to do it.
No worries. If you stop sharing, Sasha will share the other deck.
All right, perfect.
Thank you. Sorry, everybody. I'm having a technical difficulty on my end. So hello, everyone. Thank you to the EPIC series organizers for the opportunity to present. It's these types of events that set the stage for dialogue and building collaboration on advancing the science and incorporating NAMs into regulatory frameworks. As Rachel said, I'm Michael Lowit from the Environmental Fate and Effects Division, or EFED, of the Office of Pesticide Programs, or OPP, at the U.S. Environmental Protection Agency. My presentation is going to be a high-level overview of EPA's perspective on applying the 3 Rs to ecological risk assessment in support of the registration of pesticides, and will include an overview of our evaluation of the potential use of CATMoS in a regulatory context. Next slide, please. So OPP has a long-standing commitment to the 3 Rs principles, which are to, 1, reduce animal use. So for example, requiring fewer whole animal studies, replace laboratory whole animal studies by the implementation of in vitro studies, which are generally meant to indicate studies with cells and tissues, in chemical studies, which are abiotic chemical reactivity assays. In other words, cells are not used. And computational approaches, also called in silico, which include methods like the use of quantitative structure-activity relationships, or QSARs, to predict toxicity, like we'll discuss today. And finally, refine study protocols to reduce suffering. For example, this might be the modification of an existing study design to lessen or avoid animal pain and distress, or it could mean simply requiring fewer individuals in the test. There are other 3 Rs that some of you have probably heard of that are also important to our efforts as well, such as reproducible, reliable, relevant, and regulatory context. Next slide, please. An important question to ask is, why would we want to apply the 3 Rs principles to the way we currently assess risk from pesticide use? First is for reasons of ethics and animal welfare. Do we actually need whole animal testing to be protective of human health and the environment? And when is it necessary, and when maybe less so? The second reason is efficiency. Theoretically, there should be savings in both time and money once 3R strategies are adopted. The third reason is public health. This gets at the idea that it's known in some cases that test organisms aren't the best models for humans, and that in vitro studies with human tissues and associated modeling have greater human relevance and can lead to improved science. A parallel idea for protecting the environment might be using in vitro studies to test a greater number of taxonomic groups rather than simply relying on the in vivo testing with one taxonomic group, such as a surrogate for other taxonomic groups. And a fourth reason is expectations, which I'll address in the following slides. Next slide, please. So what does OPP do and why are we concerned? Are we on the, yeah. Why are we concerned with animal testing? OPP is charged with regulating the use of all pesticides in the US. The standard approach is to base ecological risk assessments on toxicity data primarily derived from whole animal studies, which are required in the Code of Federal Regulations 40 CFR Part 158 for the registration of conventional pesticides. Next slide, please. It's important to recognize that the generation of that data can be costly and uses many individual animals for the registration of a single pesticide. Costs can exceed $100 million, and the number of animals tested can be in the tens of thousands. It is critical that we work with outside partners to advance the application of the 3 Rs in a regulatory context. To that end, we are working with a wide variety of stakeholders to systematically look at the studies we require for registration, looking for opportunities to provide waivers to reduce testing and costs without negatively impacting our ability to protect human health and the environment. We're also active in work to advance the use of new approach methods, or NAMs, in regulatory risk assessment, although those activities generally have a longer time horizon for regulatory acceptance. Next slide, please. As part of advancing these efforts, OPP staff are active participants on various committees of ICFAM, which is the Interagency Coordinating Committee on the Validation of Alternative Methods. ICFAM is an important stakeholder group consisting of representatives of 17, although as told the other day, I think we're up to 18 federal regulatory and research agencies that require, use, generate, or disseminate toxicological and safety testing information. ICFAM facilitates the development, validation, and regulatory acceptance of test methods that replace, reduce, or refine the use of animals in testing. My science division in OPP, EFED, is an active participant on the ecotoxicology work group of ICFAM. And it's important to point out that the real driving force of ICFAM is NICEDM, and they provide scientific and operational support for ICFAM technical evaluations and related activities. Next slide, please. The data requirements that I mentioned in Part 158 provide the scientific basis for characterizing the potential risks associated with pesticide exposure. However, there is flexibility in implementing the Part 158 data requirements, and OPP has a long history of practicing that flexibility. Additional data can be required, but alternative approaches can be accepted and studies can be waived. Next slide, please. In 2013, guidance was released by OPP on guiding principles for these data needs. The purpose is to provide consistency in the identification of data needs, promote and optimize the full use of existing knowledge, and focus on the critical data needed for risk assessment. The guiding principles re-emphasize the flexibility in implementing the data requirements in the context of new and emerging tools, which may be used to support risk assessment and risk management decisions. Next slide, please. In 2016, the director of OPP released a letter to stakeholders reinforcing OPP's commitment that I mentioned to the 3 Rs and highlighting 3 main objectives, which are to critically evaluate the studies that impact OPP decisions on pesticide registrations, expand the acceptance of alternative methods, and reduce barriers with stakeholders and other regulatory agencies in the advancement of adopting alternative methods. Next slide, please. In 2019, EPA put out a directive from the administrator prioritizing efforts to reduce animal testing. One of the priorities of these efforts is to host an annual conference on the state of the science on the development and use of NAMs for chemical safety testing. As defined in the work plan, new approach methodologies refer to any technology, methodology, approach, or combination that can provide information on chemical hazard and risk assessment to avoid the use of animal testing. The final reports for the first 3 conferences are available on the website listed, and the 4th conference is planned for later this year. Another priority in the directive was the development of a work plan focused on the development, testing, and application of methods that will allow for the reduction in whole animal testing while remaining protective of human health and the environment. Next slide, please. EPA's NAMS work plan was created to prioritize agency efforts and resources toward activities that aim to reduce the use of animal testing. This work plan was developed by experts across the agency to set the objectives and strategies for using NAMS. The work plan was updated in December 2021 with the main objectives and strategies being unmodified. Next slide, please. The NAMS work plan identifies 5 objectives for achieving the reduction goals while ensuring that the agency's regulatory compliance and enforcement activities, including chemical and pesticide approvals and agency research, remain fully protective of human health and the environment. One of the useful aspects of the document is that it lays out strategy, deliverables, and timelines for accomplishing the objectives. However, like any roadmap, the work plan represents a snapshot in time, and this document will need to continue to evolve as EPA's knowledge and experience grows. The agency is committed to regularly reviewing the work plan to ensure the objectives and strategies, deliverables provide the best path to success. Next slide, please. So the first of the 5 objectives is to evaluate regulatory flexibility for accommodating NAMS, which seeks to determine how much regulatory flexibility exists for each EPA program requiring toxicity studies and any roadblocks that those programs may have applying NAMS for regulatory purposes. As mentioned earlier, OPP has previously identified that it has flexibility in implementing Part 158 data requirements through waivers and alternative approaches. The second objective is to develop baselines and metrics for assessing progress, and this is to determine how many animals are used in a program and track how various efforts have reduced that number. The third objective is to establish scientific confidence in NAMS and demonstrate application to regulatory decisions. Scientific confidence associated with in vivo tests comes from the decades of experience in their development and application. As we transition to use of NAMS for making regulatory decisions, we need to continually build more scientific confidence in the information from those approaches while also establishing the appropriate expectations for their performance and demonstrating their application to regulatory decisions. In part, this means that regulators will work with method developers to ensure that NAMS assays and approaches are fit for purpose and have a domain of applicability that is suitable to address the data needs of a specific regulatory context. The fourth objective is to develop NAMS to address scientific challenges and fill important information gaps. In other words, what NAMS do we need to alleviate the need for whole animal testing? The data gaps that need to be filled or the scientific challenge to be addressed is tied to the specific regulatory needs to be addressed. Therefore, a single NAM or combination of NAMs will unlikely be applicable in all need contexts. And the final objective is to engage and communicate with stakeholders. I can't stress this one enough. Widespread use and reliance on NAMs in agency decisions requires a fundamental change in thinking, given that traditional approaches have been used for decades. Given this, effective engagement and communication with stakeholders is essential to increase acceptance, obtain constructive feedback, and improve the acceptance of using NAMs to inform agency decisions. Next slide, please. There are a variety of strategic plans and roadmaps out there, and this slide shows one from ICFAM and 2 from EPA. They're generally similar, but what I want to highlight here is that each one indicates that the introduction of NAMs into a regulatory context is an iterative process, which is heavily dependent on the collaboration among stakeholders, and specifically the assay or model developers and the end users, such as a regulatory office. Early and continued engagement with the model or assay developers increases the likelihood that a particular NAM can be used in a particular regulatory context. In other words, is it appropriate and fit for purpose for making regulatory decisions? Next slide, please. So the remainder of my presentation is going to focus on the potential use of CATMoS in a regulatory context, now that you've got some background. Although I will focus on CATMoS, I wanted to point out that IFED has many ongoing projects, as well as some that have been completed and have led to guidance on potential waivers of studies. For example, in contrast to potential replacement of in vivo data with an in silico approach like CATMoS, we also have retrospective projects, meaning that we're going back through our existing data sets of laboratory studies to determine which studies have the greatest impact on risk assessment conclusions. The idea here is that we might be able to grant waivers for studies that aren't expected to have an impact. Naturally, there may be concerns that using fewer animals in testing could impact our mission to protect human health and the environment. However, it's important to stress that while OPP is committed to reducing animal testing, it is not at the expense of protecting human health and the environment. We will only move forward with any of the 3 Rs once we are satisfied that the alternative is ready for a policy change. Next slide. So this slide is to give you a flavor of the types of projects we're working on. The 3 listed retrospective analyses may lead to the reduction in the number of in vivo studies required for these taxonomic groups. We have already created guidance on this for acute toxicity to birds. Another effort led to potential refinement of the number of test concentrations needed in fish bioconcentration studies. In terms of QSARs, we have developed guidance on using the ecological structure activity relationships, or EcoSAR class program, that is used to estimate toxicity for aquatic organisms. We use that as a line of evidence for, one, determining residues of concern from pesticide use, and secondly, for determining the need for requiring additional aquatic organism ecotoxicity testing of transformation products. There's also an ongoing effort by the Office of Research and Development, or ORD, at EPA to expand the coverage of pesticide mode of action offered by EcoSAR alone, and by extension, improve our predictive capability of this approach. And finally, as will be discussed today, is our work to evaluate the use of CATMoS to estimate rat oral acute toxicity values. Next slide, please. So in collaboration with NICEDM and the Humane Society, EPA evaluated the utility of CATMoS to replace the LD50 values obtained from the acute oral whole animal tests with lab rats that are submitted in support of registration decisions on conventional pesticides, which are active ingredients other than biological pesticides or antimicrobial pesticides. Our analysis is complete, and we just published those findings in Regulatory Toxicology and Pharmacology. And I encourage you to look at the paper. As for all of the other projects I mentioned, the next step is for EPA to determine if the model is suitable for developing a waiver guidance for the acute rat oral toxicity studies, which would include under what conditions a waiver could be granted and what assumptions would be made in the risk assessment when a study is waived. Next slide, please. So before we consider the results of our analysis, it's helpful to understand how EPA uses the in vivo acute oral toxicity to rat data in the registration of pesticides. As I mentioned earlier, OPP uses data specified in the 40 CFR Part 158 to make regulatory decisions. For all registered conventional pesticides, an in vivo rat acute oral toxicity test that results in the determination of an LD50 value is performed for the technical grade active ingredient, or TGAI, as well as end product formulations. We use that data for 2 main purposes. First, pesticides are assigned to an acute oral toxicity category based on the LD50 value, ranging from category 1 to category 4. Those toxicity categories determine the precautionary statements placed on the pesticide label for acute human exposure. Second, the data are used to protect wildlife, as the LD50 value is used as a surrogate for acute oral toxicity to all mammal wildlife. Potential risks to wildlife are determined by comparing a body weight adjusted LD50 value for different size classes of mammals we are protecting to the estimated acute exposure dose of the pesticide via food items that they may encounter. Next slide, please. So this slide is just to illustrate the EPA toxicity categories that are associated with different levels of acute oral toxicity to rats. When these studies are conducted, it's common for limit tests to be conducted that simply demonstrate that the LD50 is greater than 2,000 or 5,000 milligrams per kilogram body weight for compounds the registrant anticipates are relatively low toxicity. The slide also shows you the type of precautionary statements and signal words that are placed on product labels. Next slide, please. So what is CATMoS? As Kamel already discussed in detail, most simply for purposes of our analysis and this presentation, it's an in silico predictive tool for estimating acute oral toxicity based on molecular structure. Next slide, please. So what can CATMoS do that is important in terms of a regulatory context for EFED? For our purposes, it's important that it can, one, provide both, or that it can provide predictions of both, one, the discrete LD50 values, and two, the EPA toxicity categories 1 to 4. Next slide, please. Now that we have a basic understanding of our data needs, the potential utility of CATMoS to address that data need, we can consider the project question. On the surface, it is straightforward and can be tested with existing data on in the chemical space. Our question is, how well would CATMoS perform in the chemical space of conventional pesticides in predicting acute oral toxicity LD50 values relative to the in vivo empirical LD50 values of TGAIs, or technical grade active ingredients, for pesticides registered by the EPA? In other words, can we have confidence in CATMoS estimates for adequately understanding acute oral toxicity to rats for a chemical proposed in the future for registration without the need for submitting an in vivo study? Next slide, please. So how did we do the comparison? We focused the analysis on conventional pesticides registered by EPA between 1998 and 2020, given that more recent chemistries are potentially more representative of future chemistries. We obtained the registrant submitted acute oral toxicity to rat data for the technical grade active ingredient for each of these chemicals. After data set curation, we had 177 chemicals with in vivo data to compare to CATMoS predictions. The data set curation included considerations such as removal of rodenticides, because EPA would unlikely grant a waiver for that type of chemical. The final data set had broad and relatively even coverage among herbicides, insecticides, and fungicides, with a few additional modes of action. All the data in the comparison data set are publicly available in EPA risk assessments and were conducted according to, importantly, they were conducted according to EPA test guidelines. CATMoS was run using the chemical structures of those 177 chemicals, and we considered the output of toxicity categories and discrete LD50 values, along with the 95% confidence interval. The accuracy and reliability of those predictions were assessed only relative to the existing guideline in vivo data that EPA uses for labeling and risk assessment. This is a very important point, because although open literature in vivo data might produce different results and reflect greater variability in toxicity, acute toxicity to rats, the standard used by EPA for registration is the EPA test guideline. So that's what we're trying to replace here. Next slide. First, we will look at the results of the toxicity category comparison. This can be illustrated in several different ways, as shown in these plots and the table. Ideally, we would like a high proportion of exact matches, and from a regulatory perspective, we want to guard against underpredictions of toxicity. That is, predicting a chemical is in a less toxic category than would be identified with an in vivo test. The three figures on the left show the relative percentages of matches, over predictions, and under predictions on the x-axis for different types of comparison of the data on the y-axis. Exact matches are blue, over predictions are in orange, and under predictions are in gray. The important thing to point out here is the small percentage of under predictions in gray, which is what we want to avoid. It's easier to see in the table that most of the in vivo data are in the less toxic categories 3 and 4, and that when those categories are combined, CATMoS predicts 97% of those chemicals to be in either of those two categories. This can be seen in the gray box, which shows that 145 of the chemicals identified as category 3 or 4 with empirical data, shown in the last two columns, were predicted by CATMoS to be in either of those categories, as well as shown in the far left column. 6% of the category 3 chemicals were underpredicted as category 4. From a regulatory standpoint, the overpredictions of category 4 as category 3 would be acceptable if it was also acceptable to the registrant. Next slide, please. For the LD50 comparison, it's important to recognize that this CATMoS prediction is needed for quantitative risk assessment and therefore needs to be more accurate than required for assigning acute toxicity categories on a label. As previously mentioned, CATMoS predictions of the LD50 include a 95% confidence interval, which is based on variability in published empirical in vivo data. We considered this 95% confidence interval when assessing the accuracy of the CATMoS estimates for the 177 chemicals in our dataset. Next slide, please. So this slide illustrates the results of that comparison. Each of the 177 chemicals is ordered on the x-axis from left to right in terms of decreasing CATMoS predicted LD50 values. In other words, the most toxic CATMoS predictions are on the left and the least on the right. This is shown in the orange bars. The yellow and gray lines show the 95% confidence intervals around those predictions. Paired to each CATMoS prediction is the in vivo LD50 value shown by the blue bars. In those cases, non-definitive LD50 values at 2,000 and 5,000 milligrams per kilogram are shown as definitive values out of necessity on this plot. One obvious observation is that CATMoS tends to overpredict the LD50 values. However, from a regulatory perspective, the interesting predictions are really in the black box. In those cases, a CATMoS prediction of 2,000 or higher was associated with an in vivo LD50 of 2,000 or higher in 96% of the cases evaluated. And the few chemicals with the in vivo values chemicals with in vivo values less than 2,000 ranged from 1,300 to 1,800 milligrams per kilogram body weight. This is a really important finding because there is generally greater room for uncertainty in our risk assessment for less toxic chemicals when considered relative to anticipated exposure in the environment. Next slide, please. Overall, CATMoS showed good agreement with empirical values when assigning chemicals to toxicity categories 3 and 4, which represents the least toxic chemicals. But in some cases, the predictions could result in more stringent label warnings if the predictions were used in lieu of in vivo data. A small number of chemicals predicted by CATMoS to be at the lower or more toxic end of category 3 were in fact category 2 chemicals according to the in vivo data. This point highlights the need to consider other lines of evidence before we use CATMoS to avoid this type of error. Finally, we had too few chemicals in category 1 or 2 to draw useful conclusions, and it's important to note that EPA would want greater confidence in the accuracy of LD50 values for those types of chemicals because they're obviously more toxic and making it less likely that waivers would be granted. Really, we're looking at that area, having confidence in predicting the less toxic chemicals. Next slide, please. For the discrete LD50 values, which again we use for risk assessment, estimates of greater than 2,000 appear to be reliable when considered in a regulatory context. In other words, we can have greater uncertainty about the precision of those estimates as long as we're confident that it is in fact above 2,000. For example, our screening exposure scenario for pesticides with the highest exposure potential is a small mammal that we model eating food items represented by shortgrass. In that case, up to 10 pounds of a pesticide, which is a lot, could be applied, and we could still conclude low risk potential for a non-listed species if we have confidence that the LD50 is in fact 2,000 or higher. It's okay if we don't know the exact number as long as that we know that it's 2,000 or higher. The overall takeaway is that like other NAMs, we don't anticipate CATMoS to be a one-to-one replacement for in vivo data, but rather that those predictions could potentially be used in lieu of testing, depending on considerations such as the LD50 prediction and anticipated exposure potential as in the example I gave you. These results will help us inform, from this manuscript, will help us inform possible guidance on waivers in terms of under what conditions, which chemicals, and any assumptions that would need to be made in the absence of in vivo studies if we were to use the CATMoS estimate in replacement. Finally, additional work could focus on estimating LD50 values for product formulations. So far, we have focused on technical grade active ingredient. And those product formulations also require acute oral toxicity to rat data and can show differential toxicity to the TGAI because of the other included ingredients like inerts and other active ingredients in those formulations. Next slide, please. So finally, as we consider the adoption of alternative methods, it's become apparent that it's unlikely to find one-to-one replacements of traditional whole animal test methods, whether it's CATMoS or other approaches in our experience. This means that we need to figure out how a NAMS or combination of NAMS can be used to address specific needs. And it also means that we need to recognize that the application of a NAMS in one regulatory context may not be appropriate for another. In other words, as we build confidence in the use of NAMS, we need to consider the idea of fit for purpose. And this requires not only creativity, but close collaboration between method developers and the end users. For example, a given assay might be good at predicting the lack of toxicity of chemicals in a specific chemical group, but not a precise LD50 value. From a risk-based regulatory perspective, results from that assay might be sufficient to have confidence that there is low risk for some chemicals while still needing to rely on traditional animal testing for more toxic chemicals. Next slide, please. In closing, OPP is committed to reducing animal testing burden without compromising the quality of risk assessments. This includes efforts by IFED to lead change in environmental risk assessment. Progress on incorporating reduced animal testing efforts into a regulatory context requires collaboration across many sectors, transparency, and peer review. If you have further questions, you can visit the ICFAM website and send an email to the address shown. Last slide. And finally, work like this can only be done with a collaborative group of dedicated, bright, and creative folks, bringing a wide range of expertise and perspective to the table. And I'm proud to acknowledge the folks on this team as listed here. Thank you for your time and attention.
Thank you, Michael. That was a wonderful presentation and companion for Kamel's technical presentation. They really paired well together, so thanks for that. Just a couple of notes before we get started on the Q&A, and I'll invite our speakers to turn their cameras on, as well as Amy. Amy and I will moderate the questions that are coming in. If you asked a question, some of them are being answered already. Kamel has very kindly answered some of them in writing. So you can go to the little answered tab and see some of those answers if you haven't already. I am also pleased to let you know that we have Nicole Kleinstroyer, who is director of NICEDM here. Her bio is that she's director of NICEDM and the executive director of the congressionally mandated Interagency Coordinating Committee for the Validation of Alternative Methods. And NICEDM, of course, as Kamel mentioned, is a federal resource for alternative testing methodologies and part of the IIVS. Nicole has joined to kind of join the discussion and answer any additional questions that might come up. So I'm going to start with a very general one because I think it helps us that we've got all three of you, and I think we can get some input from all three of you on this one, which is, how is the acceptance of CATMoS by regulatory authorities? And so we can see, maybe we'll start, Michael, with you if you wanted to add on to anything from your presentation, and you've just presented how EPA is thinking of applying it, and then we can ask Kamel and Nicole to jump in.
Sure, I'll just reiterate real quick, because I don't know when the question came in, that I'm only speaking for the Office of Pesticide Programs. I mean, there's other parts of EPA that you know, might be able to use CATMoS in a different context. You know, our first step was to, you know, through this partnership, you know, assess how much confidence we would have in those estimates in this in the domain of applicability of pesticides. And you know, the next, you know, now that we've done that, our next step is purely one that's internal. We're going to look at, you know, I think, you know, it was pretty obvious we're going to be looking at focusing our efforts on predictions of chemicals with low toxicity, and we're going to work out internally, you know, what factors beyond just having the CATMoS estimate will need to, in a weight of evidence, to have confidence that we can waive an in vivo study, and we're just starting to do that. So, you know, I can't really talk about exactly what those factors would be, but, you know, one of them that I mentioned clearly, because we're doing risk assessment, is exposure is going to come into it. And yeah.
I'll just add to that on the EU side, there's been a number of presentations from the European Commission, from DG Environment at various meetings over the last year. Most recently, the NAM workshop that was held at the end of May, beginning of June, on potential consideration of in vitro cytotoxicity combined with CATMoS as a QSAR prediction for the acute oral LD50. So it's not been finalized yet, but it's under consideration in Annex 7 under REACH.
Thanks, Nicole. Anything to add, Kamel?
There's one specific question here, Kamel, that got a couple thumbs up, so maybe we can start there. How can I tell if I'm inside or outside the domain by checking the AD and or confidence index?
Yeah, so like I showed in the output file, you have three columns. One is the applicability domain, and it says it's in or out, so zeros or ones. And then you have the applicability domain index, which tells you a little bit about how similar is your compound to the training set. And that's a range from 0 to 1. Obviously, the higher, the better. And then you have the accuracy, which is specific to the chemical right here. So this is the ID. It's either 0 or 1, and then the ID index is from 0 to 1, and then the confidence index, which is the accuracy. So the closer to 1, the better, obviously. There's no specific threshold for that. So just the higher, the better. Certain chemicals are easier to predict than others. So for some chemicals, you'd expect high accuracy. For others are harder. You'd be happy even with a little bit less than perfect. And also it depends on the use, like for research or for regulatory assessment, or depends on the user, what are you going to do with it. So it depends on a lot of factors. We don't have a specific threshold for that.
And maybe just tacking on to that question a little bit, there are a number of questions submitted about the applicability domain. So antimicrobials, intermediates, formulations. I wonder if you could maybe speak a little bit to those, or just the applicability domain in general.
Yeah, so well, I think here that's a different type of applicability domain. It's not. So what I call applicability domain is the implementation of a specific statistical approach that compares your chemical or the chemical structure that is being tested within the molecular, it's very technical, within the descriptors in comparison to the descriptors being calculated for the training set. So that's what I call applicability domain. In simple words, it's the similarity of your chemical to the training set. That's my applicability domain. Now there is another use outside of the QSAR world of calling an applicability domain is maybe like the types of input, which can be UVCBs, inorganics. QSARs in general are only applied to organics. That's it. It does not work for peptides. It does not work for proteins, nanomaterials, mixtures, inorganics. UVCBs don't even have a structure. So any chemical that does not have a defined structure, it cannot be predicted by QSAR, not just CATMoS or OPERA, by QSAR in general. There are other models being developed for nanomaterials, specific to nanomaterials. They're not the regular QSARs, but that's so as a group in general, QSARs are for organic chemicals.
I would even say that there's a third use of applicability domain, which tends to be used to talk about types of chemistries or even uses of chemicals, right? Antimicrobials versus conventional pesticides. That isn't really based in structure, but more in the use of a chemical, and kind of has a more of a regulatory implication or understanding. So that is, I think, even beyond, that really speaks more to Michael's presentation about how EPA is going to apply the tool, right?
And can I just jump in there and say that,
Yeah,
we had a comment or a question, several questions from Luis Valero from Center for Veterinary Medicine at FDA. And, I think that there is absolutely an opportunity here to sort of assess the performance of CATMoS in the parasiticides realm. And so, would love to follow up and see how NICEDM can support that. And just to quickly say that Kamel has already worked with the OECD QSAR toolbox developers to implement the entire OPERA suite of models, including CATMoS, in the OECD QSAR toolbox.
Great, thanks, Nicole.
Yeah, and for the antimicrobial or pesticides, or as long as it's an organic chemical and it has a defined structure, it can be predicted. And what you need to look at is those 3 columns that will help you identify whether your chemical is inside or applicable or outside the applicability domain of the model.
So we have a couple of questions that are sort of related, so I'm going to take them at the same time. And it's related to the EPA project that Michael presented. And they're asking, well, first of all, does the model, so does CATMoS include data from confidential submissions to EPA, or are they built entirely on publicly available data?
Entirely in publicly available data.
But then when the EPA did their assessment, did that use confidential submissions? No. Okay, great.
No, we intentionally, even though we might have additional data, we intentionally, to make this as transparent as possible, limited the analysis to data that's presented in risk assessments that are all publicly available. So you can't get a hold of the original studies, right? But the endpoints predicted, those are in the publicly available risk assessments.
That said, for that evaluation, like Michael mentioned in his presentation, the guidelines can be a little bit different. So the EPA guideline is very strict into what they accept as empirical value. But what we found online, in the literature, can be from different types of experiments. So we did that curation, but still there's a little bit of difference in guidelines. I don't know much about the specificities, but there are some differences.
So just also a very specific question about the evaluation. For the compounds that were used for that evaluation, was there someone that checked to see if those compounds were not among the training set compounds?
Nicole, you want to address that?
I mean, yes, the simple answer is yes.
Okay.
That's all in the
That's all we
publication.
need. So we are at the end of time. We have a few more questions. I wonder if people are okay to stay on an extra couple of minutes because we still have a lot of folks online. And just to note that the slides and the recordings will be available on the PETA Science Consortium website, which you can see in your chat. All right, I think we're going to go for a few more minutes. So Amy, do you want to go ahead with another question?
Yeah, there's an interesting question here that touches on, I think, broadly how we're validating new methods and the fact that we know that some animal tests might not be very reproducible or human relevant. So the specific question here is, have you considered cell culture or other in vitro approaches to cross-validate NAMs? Maybe said another way is, the results are basically using CATMoS to see if you predict what the rat predicted, but how do we think about that in a different way, or maybe a bigger picture way of its relevance to humans as well?
I mean, I'll take a first stab at that, and then maybe Michael and Kamel want to add on to it. So you have to strike a balance here, right? So if you're trying to, achieve as good or better as the existing reference in vivo study, you have to include a comparison to that. But one of the things that we've tried to evolve in that framework is taking a really transparent look at the reproducibility of that in vivo reference standard. And so that's why, Kamel talked about looking at the sort of the variability in those in vivo rodent oral LD50 tests when we had independently replicated studies on the same compounds. And that's what allows us to derive that confidence interval around that LD50, which is then not only used to help in the assessment of the performance of CATMoS, but also to put a confidence interval around the output of the in silico prediction model, right? So that's one thing. I mean, another thing is, I think that in some regulatory contexts, as Michael discussed, this will hopefully be a strong line of evidence in supporting a potential waiver for the necessity for an animal test. In other regulatory contexts, this might be combined with something like an in vitro cell-based assay in kind of a defined approach to, really demonstrate high confidence in a category prediction, for example. So I think there's a lot of different ways that you can incorporate the variability of the existing reference standard in both the performance assessment and the output of the model, and ways in which it could be combined with in vitro assays. I would also, we've talked to folks that are developing in vitro based acute tox prediction platforms and models, and talked about, trying to do some stress testing and look at areas where, if there's a discrepancy in what's being produced by the in vivo model and the in silico model, is there a way for the in vitro platform to kind of resolve that discrepancy? So I think there's maybe some more targeted analyses that need to be done in that space. But as both of our presenters showed today, at least in the area where the in silico model is predicting, non-toxic or minimal toxicity, very high LD50 values, it's incredibly reliable.
Thanks, Nicole. There's another question about that's specific to the output of the CATMoS model. So they're wondering if the output would contain things that regulators would like to see. So analogs used in the prediction, applicability domain, or whatever we think might be relevant for this particular model to output in order to, report on how the model was used and how, and so that the regulators can evaluate it properly.
Yeah, so the output, there are different output options, and one of them is getting the 5 nearest neighbors. So with your prediction, you get the 5 most similar chemicals to your chemical that is being predicted from the training set, or from the knowledge base of the model, since this is a consensus, not really a direct training set. And that helps identify, those can be considered as analogs and used as such. And also the similarity is endpoint specific. So it's the similarity that's relevant to acute oral toxicity, not just some random, not just general similarity, but it's specific to that endpoint.
Oh, okay. Thank you.
And maybe for a last question here, I'm wondering if anyone wants to answer this question. There's a lot of questions about commenting on just what can we do today? And, can we use it in a weight of evidence? How would we use it in a weight of evidence? Is it particularly ready for more toxic or less toxic substances? Is it more or less ready for any specific chemical type or sector? So maybe if everyone could comment a little bit on that. And Michael, maybe one kind of direct question is, I don't know if you're able to comment on this, but is EPA working on putting out any kind of notice about this, any kind of position statement or stance on the use of CATMoS in the future? So yeah, curious to just hear any comments on any of that.
Sure, I can start, and then maybe Kamel and Nicole can talk broader beyond pesticides. Yeah, so I think one of the things I was try to convey was that when we do any of these sorts of take into consider using alternative methods, we kind of have a stepwise approach, and we're at the last stage now. That second to last stage was doing some sort of analysis. It looks different for each approach, right? In this case, it's that paper that I just went through, and it was like, hey, we've already got this huge data set that we have. Let's use it. Let's see how well CATMoS can predict what we already have, and that gives us a good sense of like, hey, can we use this in the future? Because for my office, really the important thing is not old existing chemistries. It's someone's coming in with something new. They want to register a brand new chemical, and the question is, can they use this? Can they use CATMoS in some sort of weight of evidence to not give me the study that is traditionally been required? And I think what this has shown pretty clearly is that we're going to need to table the more toxic chemicals, at least right this stage, and focus on those less toxic chemicals for two reasons. One, CATMoS was really good at least showing that those chemicals were not toxic. I can't remember the percentage, it was 96% or something, right? Once you get up into those levels of toxicity, the precision just doesn't matter, because exposure levels in the environment are just so much lower that we can absorb some uncertainty there. So for our purposes, that's where we're going to focus. That's what that paper demonstrated. And so, this last step is really us internally huddling and figuring out what policy and guidance we can provide on when and where and under what conditions and what types of chemicals that we think we're going to be willing to provide waivers on. And that's what that next step is. And so yes, the intent is to put out that guidance so that when a registrant wants to come in with their new chemical, they can just literally go through it and see if it matches our expectations.
Anyone else like to comment? Okay, well, we've already taken up so much of your time, speakers and Dr. Kloensteuer, so thank you, Michael, Kamel, Nicole.
If you don't mind, I'll just add one more comment here, which is, this was a really, Michael highlighted this at the end of his talk. This was an incredibly collaborative, multidisciplinary effort across all, different organizations, agencies, NIH, EPA. But I just want to highlight the leadership of Pat Bishop from the Humane Society, because she just was a tremendous force in moving this forward, and she's the lead author on the paper. And so we all owe her a real debt of gratitude for all of the effort and the energy that she put into this. So I just would be remiss if I didn't circle back to that and say thank you so much, Pat, for all your hard work. So, and thank you to the organizers for giving us the opportunity to present this and to discuss it today.
Thanks, Nicole. Well, thank you all for listening, for joining us, and stay tuned for the next webinar, which will be in a few months. Have a great end of summer, everyone. Bye.
Thank you.
Thanks,
Bye.
everyone.